Welcome to this lesson on gene mutation. We've been talking a lot about DNA and how DNA codes for proteins, and we had mentioned at least a few times about this idea of mutation, but in this lesson we're going to go into a little more detail. The first thing let's consider is what causes mutation. You know that there could be a random error. We talked about random errors always occurring. Some of these are just spontaneous. They just happen. As the DNA is being replicated, there's just a mistake that's made. Other times, there is an increase in the number of these errors caused by something known as a mutagen. A mutagen is some substance or some thing that causes an increase in the rate of mutation. Now, there are a variety of different mutagens. There are chemicals known as alkylating agents that can do this. Any kind of ionizing radiation, such as X-rays, gamma rays, that kind of thing, or even ultraviolet radiation. These are all possible mutagens. If there's a mutation that happens during DNA replication in a somatic cell, okay, this would be prior to mitosis, then this mutation could lead to cells developing into cancerous cells. Now, there are lots and lots of fail-safes, lots and lots of things that happen that will stop this particular mutation from leading to cancer. Of course, if all of those fail, then we do get a cancerous cell, but in most cases, a single mutation will not lead to cancer. But of course, there's a much more devastating effect if this mutation happens in a gamete, such as the DNA replication that leads to meiosis, which leads to the formation of gametes. In this case, it could result in what we call a genetic disorder. It could result in a change in the entire offspring, you know, the whole genome of the entire offspring, because it's been changed within the gamete. But later in the spring, we'll talk a lot more about variation of traits, and in some cases, this type of mutation could lead to a beneficial variation in an organism, and it could actually end up being a good thing. Now, mutations, remember, are happening usually during DNA replication, and it's happening within within the DNA. But remember that DNA, sequences of DNA, are genes that code for proteins. So that's what DNA is doing. It's coding for a protein, and that any change in the protein could change anything from enzymes that are overseeing metabolism to structure of cells, all the proteins that are in the cell membrane that we talked about, the proteins that make up some of the organelles, the proteins that are involved in DNA packaging. You know, we talked a bit about those proteins. But even more than that, it could lead to a change in an entire organism. Now, the simplest form of mutation we call a point mutation. A point mutation is a mutation that's taken place just in a single base in a sequence. So you'll have a long DNA sequence, and there's a change in just one of the nucleotide bases in that sequence. This is called a point mutation. Now, in many cases, enzymes will come in and repair the error. We talked about this before, about how if there's a mistake made, a lot of time enzymes will come in and put the correct base in there. But sometimes it's missed, and the error remains. If there's a change in one nucleotide base, that means one triplet code in the DNA has been changed, which means one codon on mRNA has been changed. That means one amino acid will be changed. Now, sometimes this can have a devastating effect even with just one amino acid changed. An example of this is the disease known as sickle cell anemia. For instance, here we have the normal sequence for hemoglobin in DNA. Now, hemoglobin is a protein that's found in red blood cells, and it's the one that's carrying the oxygen, and it's pretty much controlling how much oxygen your body's getting, so it's sort of important. In the DNA, the code here is CTT, cytosine thymine thymine. The mRNA codon then would be guanine, which is going to base pair with cytosine, adenine, and adenine. And the resulting amino acid in this particular spot is GLU, which stands for glutamic acid. Now, with just a single point mutation here, we have an adenine where there was supposed to be a thymine, and so the resulting codon is GUA, not GAA. As it turns out, this is a change in the amino acid from glutamic acid to now the amino acid valine. And because of that one change, you end up with a hemoglobin with the wrong shape and sickle cells. What that means is the red blood cells can't properly carry oxygen. They have a different kind of shape. A normal shaped red blood cell is something like this. It's kind of got a little divot in the center. It looks like a donut. Sickle cells don't have that regular shape. They're shaped like a sickle, which is kind of like a little sword, and they cannot carry oxygen properly, and that of course affects every single organ in the body. So that single point mutation could lead to an effect on the whole organism in this case. Now, something else could happen, and that could be that this single point mutation doesn't really have any effect. This is called a silent mutation. In a silent mutation, because of the redundancy in the genetic code, even though there's been a change in a single base pair in the DNA, because of the redundancy, you end up getting the same exact amino acid. It's not a problem. For instance, in this diagram here, we have this sequence and then a stop codon, and the base pair change has happened right here at this cytosine. It's been changed instead to uracil when it was transcribed, and it didn't really matter because we still end up with glycine here and glycine here, and there's been no change in the amino acids. So that's called a silent mutation. There's really no effect. A more devastating kind of mutation can occur. That's called a Frameshift mutation. Now, this could be either insertion of an extra base or deletion of a base that was supposed to be there. These are called Frameshift mutations because when this happens, it changes the reading frame of the triplets or the codons. Because they're read 3 at a time, it shifts everything either to the right or shifts it to the left. So you end up with, no matter what, the entire protein is going to be changed. There's no possibility of the redundancy saving you at this point. There's going to be a change in the entire protein. So oftentimes these are very devastating kinds of mutations. For instance, here we have 3 codons. This is our normal sequence. The amino acid sequence is here. Now an insertion added a T right here, and because that T was added, we no longer have the codon CCC. Instead, we have TCC, and instead of ACT, we have CAC. So we've changed the entire reading frame, and now look, we have 2 completely different amino acids now. Same thing happens here for an insertion mutation. In this case, there's been an entire new amino acid inserted, in this case phenylalanine right here, but because it was 3 bases added instead of a single insertion, you end up without a frame shift. Instead, you just have an added amino acid. So all different kinds of things can happen when bases are inserted. This is called a frame shift mutation. And then finally, the last kind of mutation I wanted to talk about is a nonsense mutation. Nonsense mutations result in a mutation from a codon that would code for an amino acid to a codon that actually codes for stopping. So there's a mutation that happens where the bases are changed. Perhaps it's maybe a single point mutation, but instead of coding for a different amino acid, instead it just codes for a stop codon and the whole thing stops. So that means that it cuts off the protein before it's finished. Here's an example of that. We have the original DNA code here for an amino acid sequence. You see it's a whole bunch of glutamines here, one right after the other. And we have a single nucleotide that got changed here. It was, it looks like a C, and now it was changed to a T. And because it changed from CAG to TAG, that's a stop codon, and the whole thing stops right there. Instead of making the entire protein, it only makes a short polypeptide of 3 amino acids in this case. This is called a nonsense mutation. So here's a summary of the different types of mutation. Here's our original code. It starts with AUG, which codes for methionine, and then here's the rest of your code. In the normal we're using a sentence here to demonstrate how the change occurs. You know, ordinarily it would be amino acids, but this way you can kind of see what's happening a little better using words. So in this case, let's say that this code codes for DNA has all you can ask for. With a silent mutation, perhaps the U is changed to an O here, and you would still pronounce it the same, so we would call it a silent mutation because the information is still the same. DNA has all you can ask for. With a missense mutation, or a mutation that changes the amino acid, we now change the U here to an L, and it goes DNA has all Lou can ask for. That changes the meaning. A missense mutation changes the meaning. A nonsense mutation, remember, cuts it off too early. In this case, DNA has all you, and you don't get the rest of the sentence. And then finally, a frameshift mutation where there has been a change in the reading frame. We've added a Y here. Instead of ALL, it's ALY, and now you can't really even read the sentence at all. So you see what happens with each of these different types of mutation. Finally, the last thing I wanted to talk about is something really kind of interesting. There's a scientist known as Barbara McClintock who actually received the Nobel Prize for her discovery. She studied the striping pattern in Indian corn. It's a different type of corn that produces kernels of different colors. But if you've ever seen Indian corn, you'll see that it has this striping pattern, and she was studying how that could be. And she discovered these things called transposons. Transposons are actually pieces of the DNA that she discovered spontaneously move around in the genome. They sometimes move from one spot to another on a chromosome, and they've even been seen moving from one chromosome to another. So these are movable pieces of DNA. I know it sounds strange, but these are just sections of the DNA that have a tendency to move around. What they do when they move around, we'll talk more about this in class, but when they move around, they affect the DNA that's near them. So remember, genes go in a particular sequence. They could move in and interrupt the gene sequence, or they could just insert themselves in a place that affects the transcription of that gene. So either makes it transcribe more or makes it transcribe less. So they could inactivate the DNA or increase activation of the DNA. And they are known to cause mutation, these transposons. So here's a diagram of that. Here we have this transposable element. This is a transposon here, this little blue piece. In this case, it copied itself, or it was copied, and it inserted itself over here in the center of gene F, and it interrupted and became no longer functional because of that. So that's an example of transposons or transposable elements. So that's it for this lesson, and have a good day.